Hello and welcome to Badger Talks Live, which brings exciting happenings, resources, and talents from your UW flagship campus to the people of Wisconsin and beyond. I am Mara Gamara from Tigray, East Africa. I'm a fourth-year graduate student in the Cell and Molecular Biology program. My research in Dr. Mehdi Kabaja's lab in plants pathology department focuses on understanding the process of death in fungi. We hope that elucidating this process will help us combat fungal pathogen of animals and plants. It is my pleasure to introduce Letters and Science Mary Herman Rubenstein and Vilas Distinguished Achievement Professor of Botany and Bacteriology, Dr. Anne Pringle today. Today, Anne will be sharing with us the work being done in her lab. Anne's lab uses fungi as tools to test and elucidate general principles of ecology and evolution. She has also focused on the problems posed by invasive fungi. Anne Pringle obtained her bachelor's degree in biology from the University of Chicago and received her doctoral degree in botany and genetics at Duke University. She continued her postdoctoral training as a Miller Fellow at the University of California, Berkeley, where she began work with the death cap fungus. In 2005, she joined the Organismic and Evolutionary Biology Department at Harvard University. For her passion towards scientific teaching and mentoring, she was awarded at Harvard University the Fanny Cox Prize for Excellence in Science Teaching and the Mendelssohn Excellence in Mentoring Award. In 2015, Dr. Pringle moved to the University of Wisconsin, where she continues studying the complex kingdom of fungi. Please welcome Dr. Anne Pringle. Give me 30 seconds and Dr. Pringle will be right here.
Thank you again. Great. Thanks so much, everyone, for tuning in, for being here to listen to me talk with you today about something that maybe a lot of you have never thought about before. And these are the problems of invasive fungi. And fungi are very much in the news right now for lots of different reasons. Also, it's the season. I'm talking to you in October to collect fungi. So it seems like a really great opportunity to talk to you about maybe something rather different. So I'm going to take you all over the world today, including the state of Wisconsin, and we're starting now high in the Andes Mountains in the country of Colombia, and we're going to go back to Colombia in a little bit. But first I want to talk to you about one of the reasons why fungi are so incredibly popular right now. And they're popular because there's an idea that a lot of people are talking about that fungi are going to heal our planet, that they're really going to help us solve all kinds of problems. And some of that feeling, some of that thinking comes from experiments which are affectionately called the big plant, little plant experiment. And so in the big plant, little plant experiment, which has been done by me, here you're looking at a photograph of the work I did during my PhD, but it's also been done by hundreds, thousands of times by scientists all over the world. In this experiment, you grow plants with and without mycorrhizal fungi. And mycorrhizal is a really strange word, but myco just means fungus and rhizal means root. So fungus root fungi is perhaps the way to think about them. So people grow plants with and without these fungi and with different species of fungi. And when you run this experiment, what you find is that plants with mycorrhizal fungi, here you're seeing two plantago plants on the right, growing with a species that used to be called Aclospora mellia. Plants with those kinds of mycorrhizal fungi grow better than plants without any kind of fungi. And those are the control plants that you're seeing on the left. So big plant, little plant experiment. And this is a quite robust result. As I said, it's been replicated many times over. So this is part of the idea that fungi are a good in our world. They help plants to grow. Here's the same kind of big plant, little plant experiment, But this time, instead of little weedy plants, you're looking at trees. You're looking at oak trees. And on the right, again, you see oak trees growing with fungi, with truffles in this case. And on the left, you see oaks growing without truffle fungi. Big plant, little plant. Fungi help. One nuance to this, potentially, is that actually there are different kinds of these fungus root fungi, different kinds of mycorrhizal fungi. So there are endomycorrhizal fungi that grow inside roots, and these are a kind of mycorrhizal fungi that grow with, for example, your tomato plants. And there are ectomycorrhizal fungi that grow outside of roots. This is what you're seeing on the right-hand side of the screen. And these kinds of mycorrhizal fungi grow with trees. And you can't put an ectomycorrhizal fungus with a tomato plant and expect it to grow. It won't grow. Nor can you take these endomycorrhizal fungi and put them with your oak plant. It won't work. So there are different kinds of compatibilities. What kinds of mycorrhizal fungi you use to grow your big plant, it really matters. So these are things that are important to know about if one is wanting to use mycorrhizal fungi in your garden, for example. So with that idea in mind, the big plant, little plant experiment, mycorrhizal fungi can help. I want to take you first to the Southern Hemisphere. And in this photograph, we're driving through New Zealand, and I literally took this photograph out of the passenger seat of a car in New Zealand. And in New Zealand, as is true across the Southern Hemisphere, pines are not native, which is remarkable if you didn't know that before. When I first learned that, I was really surprised. Because if you go to a place like New Zealand, you see pines all over the place. In South Africa, you see them everywhere. In South America, you see them everywhere. But they're not native there. They're planted there. And why are they planted there? And you can tell, like, here are the pines in this photograph. And, you know, it's not often that you see a forest that grows as a square with neat little rows of trees, but that's what you're seeing here, these pines. And so what's going on? Well, they're planted as a crop. In the same way that you would plant soybeans or corn, these trees are being planted very specifically to be harvested and turned into products. So why is this happening? It's happening because we need things that are made out of trees like pines. We need toilet paper. Certainly toilet paper is something that's been a lot on our minds in the last year and a half. So the economics are driving this such that there's a demand for things like paper and toilet paper. So you plant the trees and you harvest them and you turn them into those products. So pine trees are not native to the Southern Hemisphere, and there's a really interesting story related to mycorrhizal fungi that has to do with pines. So here you're looking at the continent of Africa. And when pines were first planted in places like Southern Africa, they didn't grow. The crops failed. And foresters, and this would have been in the 30s, 40s, 50s, were trying to figure out why their crops were failing. And it turns out they were failing because the pines, when you planted them in Southern Africa, didn't have the right kind of mycorrhizal fungi. So foresters figured this out when they started importing soil into places. So you can see, for example, at the bottom of this presentation, there's an arrow saying and before the arrow is the word the Netherlands. A person named Nicola went around and talked to foresters all around the world. And when he talked with them, he would say, how did you get your pines to grow? And they would say things like, well, I talked to a buddy in the Netherlands and he mailed me a tin of soil. And when he mailed me that soil, I put it with my pine trees and then my pine trees started to grow. So it was the movement of pine-associated ectomycorrhizal fungi, not native to the Southern Hemisphere, like pines, not native, but moved to the Southern Hemisphere, that enabled those pine trees to grow. And Nicola recorded a massive movement of soils all across the continent of Africa, and the same dynamic was happening in places like New Zealand. Foresters were bringing in soil, but what they were really bringing in was mycorrhizal fungi to help their crops to grow. So unsurprisingly, a lot of different kinds of genera of fungi were introduced across the globe with the movement of these soils. So now you're looking at a list of names, and these are names of genera of fungi starting with Inoculate, a fairly rare genus, Amanita, a more common genus. And the black bars and the gray bars are just suggesting to you the number of times that any particular genus was found growing in a place like South Africa or New Zealand where it wasn't native, but a forester had brought it in the context of planting pines. So a lot of fungi were introduced across the globe. And here's a different way of looking at those same kinds of data. You can look at any given country, and the size of the green circle will tell you how many species have been introduced to that country. So you can see, for example, in the United States, there were introductions sometimes for different reasons, not having to do with moving soil to plant pines, because obviously the United States is in the Northern Hemisphere. But you can see in the Southern Hemisphere this massive pattern of lots and lots of species of fungi that have been introduced. So unsurprisingly then, sometimes when you're moving huge swaths of biodiversity around the globe, the next thing that happens is that sometimes some of these fungi become invasive, and that means that instead of just potentially staying nicely contained within the crop, within the pine trees, they actually start to spread outside the pines, and they grow into native forests. And what you're looking at here is a photograph of a picture of a species that I work with a lot, the death cap. The death cap is deadly poisonous. It's a mushroom that will kill you if you eat it, and it's now invasive in California. Trees were imported to California. In this context, the story may be that the trees were imported to support a domestic cork industry. Corks are harvested from cork oak. And as those trees were moved, they were moved with soil, and that soil housed spores or individuals of death caps, and the death caps spread through the forest. So that's one example of an invasion that followed movement of soils. Another, now I told you I was going to take you around the world. Now I'm taking you to Colombia in South America again. And here what you're looking at, again, we're back high in the Andes Mountains, and you're looking, this is the southernmost distribution of oaks in the world. You're looking at Quercus humboldtii. You're looking across the valley, and the trees that you see across the way are Quercus humboldtii, these really lovely endemic forests of oak. But when you go to these endemic forests of oak, what you see is this crazy famous mushroom, like the Super Mario Brothers mushroom. It's exactly that. It's the European fungus, Amanita muscaria, that's now invading in the country of Colombia. So we've gone from California to Colombia. And on the right-hand side, you're looking at a map of Colombia. And in gray are the two famous mountain chains, the Andes, within the country of Colombia. Green are places where you can find this European Super Mario Brothers mushroom in association with tree plantations, with trees grown as crops. But in blue is the start of where muscaria is now associating with these endemic native oak trees. And this is a close-up of the site where muscaria is now starting to invade. In purple and in yellow are the Super Mario Brothers mushroom growing with a pine and an acacia, and in white is the mushroom growing with Quercus humboldtii. So you can see how this fungus is likely using spores to hop from place to place and spread across the landscape. And this is an image from Google Earth. So, and here's a photograph of me up in the highlands with Natalia Vargas, who is the lead researcher and did this work. And my big joke here is that I didn't Photoshop the mushrooms in. If you look to the left of this photograph, you'll see this crazy collection of mushrooms. And it, I wouldn't blame you if you looked at those and you thought, no, surely she just stuck those in with some sort of fancy PowerPoint tool. But no, that's real. They're enormous. Death caps in California also, by the way, are enormous, bigger than they are in their native range. So we have this dynamic now of invasion, of things spreading, things that are being taken to a place where they didn't grow before, and they're spreading. And unsurprisingly, given what I told you about trees being planted as crops, the numbers, the amount of planted forest in any given country, now you're looking at lots of countries in South America, Brazil, Chile, Argentina, etc. And how much planted forest you have, which is the line on this graph, represents amount of planted forest, is correlated with how many introductions there are, which you can see in the sort of jaggedy line bars, and the dark black bars are just the European Amanita muscaria that we've been talking about. So there really is this element of human trade and human consumption and human need for paper products that's associated with the introductions of these fungi. So I hope by now that I've convinced you that there is a swath of biodiversity that's being moved across the globe by humans and is spreading. I also want to point out that this is a Wisconsin story. So it's not just a story of California and Colombia, New Zealand. It's a local story. And here's probably the most important thing I'm going to tell you today is wrapped up in these next 3 or 4 slides. So here are photographs of Hoyt Park in Madison. It's literally a 20-minute walk from my building. And you're seeing a tree far away and close up. And what's growing on this tree are golden oysters. Now, unlike every fungus I've talked about now, these are not mycorrhizal fungus. They don't help trees to grow. These are decomposer fungi. So these are the kinds of fungi that break apart trees. They bust them open. Sometimes they kill the trees, or they may attack after the tree is already dead, but they bust it apart. And in general, decomposer fungi are an important part of our ecosystem. But golden oysters are not native to Wisconsin. So these golden oysters are native to eastern Russia, China, and Japan. We know that. That's where they're from. They've been described there for hundreds of years. Now we're seeing these golden oysters appear all across Wisconsin, and not just Wisconsin, but the Midwest, and you can see on the East Coast as well. What you're looking at are just little flags that mark places where people have found golden oysters. And golden oysters are cultivated. They're delicious. They're yummy. People buy kits to grow them. And what's happening is that this fungus is escaping cultivation, and it's spreading into local forests. And now we're finding it not just contained in gardens, if you will. It's spreading rapidly outside of cultivation. And I want to point out that the research that I'm showing you was the master's thesis of Andrea L. Bruce, who worked at the University of Wisconsin-La Crosse and is now working for Bolt, which is a company that makes mushroom fabric. Again, back to the theme that fungi can offer us lots of solutions. One thing that you can do with fungi is make this amazing fabric out of it. And if you want to know more about that, check out Andrea Bruce's website or check out Bolt's website. But before she worked for Bolt, she was tracking this golden oyster that was moving across Wisconsin. Here's a sense of how much more common it is now than it used to be. So in 2014, you didn't see it. So these are counts of how many times people have seen golden oysters, both from a website called Mushroom Observer and from a larger biodiversity inventory database called GBIF. And in 2014, these are just counts from the state of Wisconsin. In 2014, it wasn't seen, and look what's happened by 2021. And Mushroom Observer, we could talk about why that pattern is more flat, but the blue one is the one I would trust more. It aggregates more data. And it's really common now. And in fact, maybe you've seen this yourself if you're a mushroom, if you go out and harvest, maybe you've experienced this directly yourself. I certainly am experiencing this directly myself. It's everywhere now in places that I never used to see it before. And when you see it, there's a heck of a lot of it. Not just a few mushrooms, but pounds and pounds and pounds and pounds of it. So, and here's the evidence that it's escaped cultivation again from Andy Bruce's master's thesis. You're looking, every dot is a collection, and the dots that are closer together are more closely related than the dots that are further apart. And pay special attention to cluster 3. Cluster 1 and cluster 2 are from commercial strains of the mushroom, and cluster 1 and cluster 2 don't appear to be related to those collections that are being made in nature. By contrast, look for the number 2 and the number 5, look for the number 2 and the number 5. Those are the same commercial strain from different places, and those are very closely related to what you're seeing in nature. So it's escaped cultivation. Please know about it. Please don't grow it. Please don't buy kits that are golden oyster. Please be thoughtful about that. If you do grow it, do not compost it. Put it in the trash. Kill it. If you see it, do pick it. Eat it. It's delicious. It's an emerging story that's happening right now in our state. This fungus that's spreading. So why are invasive species a problem? Why I started this talk off as the problem of invasive fungi, and you might be wondering by now why. Who cares if they spread around? Well, to understand that, the first thing that you need to know is that we haven't named or identified most of the globe's fungi. So using various techniques, we think we've described something like 100,000 of probably what's likely to be about between 1.5 and 10 million species. And just the fact that I'm sitting here telling you there may be 1.5 million species of fungi, or there may be 10 million, I mean, the fact that I can give you something more precise than 1.5 to 10 million should maybe suggest to you how little we know about this kingdom. There are very few people who study fungi compared to how important they are and how many species there are. So we don't have names for most of the fungi in the U.S., most of the fungi in Wisconsin. We just don't have names for them. We don't have them. So because we know so little, you get to statistics like this. So we have this sense that biodiversity is imperiled, not just in the US, but everywhere. And we try to keep lists of what's in trouble, right? We try to understand what's endangered, what's becoming rare, what's under threat, what's disappearing, whether there's a new disease for a bird, etc. So when you go to these lists, you see, for example, that for plants, there are 18,400 species and 15% of them seem to be in trouble. For fungi, there are 37,800 described species in the US, which, as I just said, is kind of a ridiculous underestimate, and only 0.4% of them are imperiled. So there's a real disconnect in the data between what seems to be in trouble for plants and what seems to be in trouble for fungi. And there are kind of two conclusions you can reach. One, fungi are less likely to be in trouble than plants, or we know so little that we don't even know what's in trouble. And I tend to think that it's the second. We know so little that we don't know what's in trouble. Things might be going extinct around us before we even had a chance to describe them. And invasive species are certainly a part of this conversation. And when I talk about this, I like to bring up garlic mustard, because I think a lot of people have experience with garlic mustard in the state of Wisconsin, and pulling garlic mustard and understand that it's a pest, right, from pest to pesto, if you have ever cooked with garlic mustard. So, and the reason garlic mustard is a problem is because it crowds out other plant species. It actually, in case you didn't know, it kills fungi. So it secretes these chemicals in its roots, and it kills the mycorrhizal fungi around it. And the thought is that that's one mechanism how it crowds out other plants that need mycorrhizal fungi can't grow when garlic mustard is present, because garlic mustard has killed the mycorrhizal fungi. So the part of the conversation of the world in trouble, biodiversity in trouble, is invasive species, and it's quite likely that things like the death cap, which are invasive in California, are also causing harm. But there are so few people who study it that I can't nail that down for you. I can give you this very suggestive kind of data, for example. What you're looking at here are, my graduate student, Ben, went to 2 forests in California, and for an entire season, he collected all the mushrooms. And then he said, okay, what proportion of the biomass is death cap mushrooms? And look at the data for Forest 2, for example. Fully 80% of the biomass, that means every single mushroom he harvested, all of them, 80% of that biomass was death cap. So that kind of, so it's a pretty icky, I mean, it's pretty icky when you're walking around in the forest. It's the forest of death. So this kind of dominance doesn't suggest that all is well. And there are a couple of different kinds of impacts death caps may have on surrounding landscapes. But it's certainly not the dynamic that you see with death caps in its native range in Europe, for example. All right. So now I want to get to a solution, potentially. So I've just talked to you about trouble. I've talked to you about different kinds of trouble. I've talked to you about, why we think it might be trouble to have invasive fungi. So what are things that we can do in the state of Wisconsin, anywhere? And to talk about that, I want to take you to the city of Bogota. So now we're back in Colombia, where the Super Mario Brothers mushroom is invasive. So we all have a sense, I think, that we want to live in spaces that are green. We want trees, we want wildlife, we want greenness around us. So that might lead us to a conclusion that sustainable, livable cities are green. And you're looking at the city of Bogota, which is a mega city. It's enormous, and we're up on a mountain, looking out over the city. It stretches all through the valley around around corners and twists and curves. It's a mega city. And like in other places in Bogota, urban trees offer healthy public spaces where you can go walk, recreational areas. Lots of us like to go throw frisbees in parks where there are trees, or have picnics, or see opera in the park if you're in Madison. Ecosystems for animals. Often we see interesting, cool things living in these, with the greenery, and if we're in a city. And then trees also minimize the wind. They shelter us from the sun. They mitigate noise. So there are lots and lots of reasons why we like to plant trees in cities and around us. So the question, and this work was funded by National Geographic, is keeping in mind the problem of moving soils, from the Netherlands to Southern Africa, and keeping in mind that we want urban trees. Can we help trees to grow the way that we help the pines to grow, but without moving soils from the Netherlands to South Africa? So that was the question, and the big idea. And here's this poor street tree that definitely needs help. It needs something if it's going to make it through another day. If you look at that picture behind the cage that it's in. So our big idea was to not take soil from far away, but instead use whole soil from a nearby healthy tree that's growing well to potentially inoculate these struggling street trees and see whether we can get this to work. And although this is a very specific application, urban trees, the idea here has a lot of potential to be used in lots of different kinds of contexts and not just urban street trees. So this is the big idea that we've been testing. I've been testing with my collaborator, Natalia Vargas. You saw the photograph of her earlier, that we've been testing around Bogota. And this is like a fecal transplant. Some of you may have heard of this. People who are very ill in the hospital with a particular kind of infection caused by a bacterium that we call C. diff, often recover if they're given, I know it sounds crazy, if they're given healthy poop from really just about anyone. Something about the microbiome, about the bacteria and maybe even fungi within that poop restores their health, and it seems to be a much more effective cure for that disease than anything else. So maybe we can cure this tree by also giving it a soil transplant. So we've been working primarily with Quercus humboldtii, which is the same tree that you saw high in the Andes, because it's very, very, very commonly planted as a street tree. It's planted both in these surfaces that are like median strips, like just a little box around the tree. And it's also planted in parks. On the right, you're seeing myself and my students sampling in a park setting. So we're working primarily with Quercus humboldtii, and we're doing an experiment where we take native soil transplants from, here this image is a very beautiful forest, but we're really actually taking them from nearby healthy trees that are just a few meters away from the trees that we're working with. And we put that native soil in with trees that are, and we have the great cooperation of the Bogota Botanical Garden. They've been wonderful helping us, and so we're actually using trees that are actually going to go out and be street trees in the city of Bogota, and we're giving them these whole soil inocula just from nearby places to see whether that whole soil transplant helps them to grow. So that's the question. And so we have 4 kinds of treatments, and this experiment is very much in progress. I'm going to show you preliminary data. We'll see over the long term whether they hold up. So far looking good, but I just want to emphasize that these are very preliminary data. So we have a control that we don't do anything to, and we have native soil, that's the transplant, fertilizer, because that would be what a lot of people would do if they were growing up street trees, and then the native soil and the fertilizer, just to make the experiment complete. So we're also doing the experiment with a native pine. That's actually not a pine, but they call it pine. but it has nothing to do with pine, but I'm not going to talk about those data today. I'm really going to focus on the oaks. So those are the treatments, and this is a picture of the nursery called La Florida in Bogota. And here's a photograph of us taking native soil. So you can see it's actually a large, healthy Quercus humboldtii at the nursery, and we're actually digging up the soil, and that's the soil that we used for our whole soil transplant. So what happened? Well, something surprising happened. And we're not 100% sure we understand it, but we have a hypothesis as to what's going on. So it turns out that the whole soil transplant is a really good idea. It works. But actually the very best thing that you can do for these street trees is add both native soil and fertilizer. And this is a result that we started seeing early on, and we're taking all kinds of measurements on, so how you're looking at the height of the tree and the diameter, the thickness of the stem here, and we're taking this at 0 days, 9 days, 97 days, 300 for the experiment has been running for over a year now. And in almost every case, what we're seeing is that the purple, which is the native soil and the fertilizer, the purple is giving us much larger plants than anything else that we could try. So why? I'm going to go back a slide. We think what's going on is that when you add the native soil, it's helpful. It's better than the control. But when you add the native soil and the fertilizer, you've basically put resources into the pot, that's the fertilizer, that the fungi can find and bring to the plant. If you just have the fertilizer, the plant can't absorb all of it. It doesn't find all of the fertilizer. So fertilizer by itself is not as good as fertilizer plus the whole soil transplant. Because when you've got fertilizer and the whole soil transplant, you're enabling the fungi to find resources. You've given them resources to find and bring back to the plant. So that was a bit of a surprise for us, but we think that that's what's going on. All right, so now, so maybe this is a solution. Maybe this is an idea. We can use native soils, which don't have fungi from far away in them. So by definition, if you're using native soil, you're not introducing foreign species. You're using native fungi. And we are wondering whether this is really a potential solution for this issue. So just to take a step back, fungal introductions lead to invasions, and by now I think we've documented lots of invasions. The spread of mycorrhizal fungi is historic. I showed you that the continent of Africa and historical data about that, but it's also ongoing. And I would say at this point that particularly if you're going to a garden store and you're purchasing a product that has a biofertilizer or a mycorrhizal fungus, you don't know where that fungus came from. Usually that fungus did not come local, is not generated locally. Typically those biofertilizers are made far away from wherever you are, and they don't necessarily use fungi that are native to where you are. So moving fungi is historic, but it's also ongoing, and there may be viable alternatives. And I just want to be really clear, I'm not telling you at this moment to go take soil from the prairie next door and add it to your prairie. I'm just saying that it's time to start the conversation. And as I said, we have a long way to go with our work at the Bogota Botanical Garden to prove to ourselves and then to the world that this is really going to work. But I do think it's time to start the conversation about fungal invasions, fungal biodiversity, and how we think about the fungal kingdom and how we use it in our gardens, etc. I want to thank Natalia Vargas. We started this experiment at the Bogota at the nursery just at the start of the pandemic, and we could have lost it all, and we didn't, because she kept on going with her masks and shields, etc. So I wanted to thank her, and I also want to thank my lab, who's been super supportive of this work. And here's a lovely dorky photograph of mushrooms that we all rode on to represent ourselves at a fungal foray not so long ago. And if you're interested in fungi, I worked hard to put together this set of 3 public talks that are now housed at iBiology. So if you want to see more, including just a basic introduction to fungi, this is a good place to look. And with that, I'll say thank you so much, and I would be happy to take any questions.
Great, Anne, thanks so much. And you really put into perspective a common household food, right, that we see all the time, and you've now put it into this foreign world for many of our viewers, I'm sure. We do have some questions posted here. Case, I'm going
to go to a more beautiful picture if I can.
Oh, yeah, please do.
Well, I guess I'll just put on there. That's nicer than the other one. Okay, yeah, I'm happy to take questions.
Thank you. Case your Foss, are other varieties of commonly cultivated mushrooms at risk of becoming invasive, like the golden oyster mushroom?
I mean, it's a great question, and I've been really so just to say also that this golden oyster thing really hit me like an anvil over the summer as I started to see it and really understand what's going on. So I don't know of anything that's quite like that right now. Are there risks? There are always risks. Some cultivars have been cultivated for a really, really long time and don't ever seem to have escaped. And I'm thinking of shiitake. So at this moment, I would say I don't see any risk in growing shiitake, for example. It doesn't. I mean, I know people have been doing that for tens and tens and tens of years in Wisconsin. So yes, I think there's definitely some that are more likely to be trouble than others. Yeah.
So this is leading up to another question that was posted, but you talk a lot about this man-made transfer of fungi, and then you also mention perhaps like spores can float in the air or whatnot. But are animals or insects also not responsible for any transition, or is that possible?
Oh, it's definitely possible. That's a great question. There are lots of different kinds of fungi that are eaten by mammals, and it's a story that I'm not involved with, but I know, for example, in South America, there are some kinds of fungi that are spreading specifically because mammals dig them up out of the ground. They're called truffles. They're not the same truffle that you might buy and grate over your scrambled eggs, for example. So it's not that truffle, but there's another fungus that makes a truffle-like shape that's very appealing to mammals, and mammals smell it and dig it up and they eat it, and then they run to a different part of the forest and they poop it out and it spreads that way. So yeah, and then of course there are other kinds of fungi that are spread by insects. So it's definitely insect vectoring isn't a part of the story for at least as far as I know. With golden oysters, I might not know one way or the other, but it's not a huge part of what I'm telling you about today, but it's definitely a huge part of the story with fungi, definitely, and super interesting.
Great, thank you. So comment here, this is incredible. I've noticed some pretty vast network of fungi in the deadfall area up north. I'm wondering about how much of their presence is due to human activity. What you're saying is...
I don't know. What I would say in response to that is that I'm curious too about those kinds of questions, and the very best thing any of us can do is start paying attention to our local biodiversity now and start recording it. There are some really fantastic websites where you and I can go and record our observations. Mushroom Observer, I'm really involved with Mushroom Observer. Of course, there's also iNaturalist, and those are two places where right away you can make an account and you can see mushrooms and you can snap pictures of mushrooms and you can post them. On Mushroom Observer, which I know the most about, you can take a picture, and even if you don't really know what it is, if you put it up, other people will chime in and give you suggestions as to what it is. So it's also very satisfying if you are just trying to figure out what's in your local landscape. Of course, you talked about networks, and of course you can't ID a fungus from its mycelium, its body, the hyphae that you see in the soil, those networks, which is frustrating and part of the reason why we don't know a lot. Sometimes I feel like we don't know a lot. So you have to have a mushroom. You have to have a spore-bearing structure. But anyway, the very best thing we can do is start recording data now. Baseline data is what it's called. So we can know what's here now to compare against the future. And it's not too late to start doing that. It's imperative that we start doing that.
Great, thank you. So as a consumer, a large consumer of mushrooms, I've wanted to dabble in foraging, but I've always been told that there are so many poisonous mushrooms that resemble edible mushrooms. So as you're talking about this golden oyster mushroom, I of course am like getting my basket ready. I'm ready to go. I would love to grab some. Is there a poisonous counterpart that we need to be like aware of in the area?
So I am going to give an answer to that question, but I'm not going to give a satisfying one. And I've stopped giving satisfying answers because I'm really startled sometimes by what happens, and you only need to be involved with one death from eating a mushroom before you become a very, very cautious person when you're talking to people about how to do this. So first, I would say if you can tell the difference between cilantro and parsley, you can tell the difference between an edible and a poisonous fungus. However, I would never try to describe to you over a Zoom call the difference between cilantro and parsley. I would try to bring you cilantro and parsley and teach you the difference between them, right? So in the same way, you have to just hang out with some knowledgeable people for a little while and say, what is this? What is that? I can't think of anything that looks like the golden oyster that you would confuse it with. But see, just because I think that doesn't mean that someone wouldn't actually confuse it for something. That's really the tricky part. So you really have to kind of experience it directly with someone, and then. But sometimes it doesn't mean you have to drag them with the woods to you. You could just bring a basket of the golden oysters you harvested to someone, or to the Madison Mycology Club, or to the Wisconsin Mycology Club, or there are lots of places in Wisconsin that have enthusiasts around. So you just need to find those people. You can find them on Mushroom Observer, as a matter of fact, and then teach it to yourself, and then you'll know, and you're fine.
Excellent, thank you. And we do have a question saying, could you please mention those mushroom sites again?
Yeah.
And we can post them in the chat as well.
Okay, great. One is called, if you just Google Mushroom Observer, that's one. That's my favorite. And then the other website is iNaturalist. And iNaturalist is not just for fungi, it's for anything. Great. Unfortunately, we don't yet have, for plants, there are a lot of really good apps that you can get where you can snap a picture of the plant and the app will tell you what plant it is. That relies on fairly complex machine learning algorithms, and we don't have them yet for fungi, but I think they're coming.
Okay, great, thank you. Well, lots of nice comments out here. Excellent, Anne, thanks so much for your time and thought. This was just fascinating. You've really shed a new light on this topic for so many of us who aren't in your world of research, and thank you for the work that you're doing. It looks like there is just an enormous amount of questions that need to be answered, and thanks for your start on that.
I really appreciate everyone's time. I know people take time to listen to these things, and I appreciate that very much.
Great. Thanks so much, Anne. Really appreciate you joining us.
Yeah.
So please tune in this Friday. We're going to be having a special Badger Talks Live as we're celebrating UW Family Weekend. And we're going to be talking with Professor of Political Science, Barry Burden. He's going to be giving us a look under the hood of the map making process and discuss the wide variety of ways that states do redistricting and how laws and court decisions do or do not keep gerrymandering in check and what can be done about it. So that's a special talk this Friday at noon. Then we'll continue our focus on the Wisconsin Science Festival, which is coming up October 21 to 24 with dozens of events, probably over 100 around the state. Visit wisconsinsciencefest.org to see what events are happening virtually and near you. We will continue the Science Festival focus next Tuesday, October 12th. We're going to be talking to Larry Shapiro and Steve Nadler, who are two professors of philosophy on our campus. They're going to be giving a Wisconsin Book Festival preview event called When Bad Thinking Happens to Good People. And they're going to be presenting their argument that the best antidote for the current epidemic of bad thinking is the wisdom, insights, and practical skills of philosophy. So for sure going to be an interesting conversation. Then the following Tuesday, we will close our Science Festival focus by talking to the co-founders of the Science Festival, Laura Heisler and George Tsugros, who will be talking about the Insider's Guide to the 21 Science Festival and how you can get the best experience and participate in those events. Please visit us at badgertalks.wisc.edu, where you can see our new podcast. We'll be having a launch of the podcast for Science Festival on October 12th with Laura Heisler, so please tune in. See our upcoming schedule of live talks. Sign up for our email list. Please consider a donation to Badger Talks. We are supported by a grant. And you can also search the roster of over 400 UW faculty and staff who have signed up to generously donate their time for free talks at locations around the state. So feel free to invite speakers to come to your neck of the woods for issues that are important to your community. We'll look forward to seeing you on Friday, and thanks for tuning in.